My name's Afshin Beheshti. I'm currently at University of Pittsburgh. I'm the director for the Center for Space Biomedicine, also associate director at McGowan Institute for Regenerative Medicine, and also have a professorship in the Department of Surgery and the Computational Systems Biology.
And hi, I'm Chris Mason, a professor of genomics, physiology, and biophysics at Weill Cornell Medicine. And we have a laboratory that also spans work at the Cancer Center, work with the hospital, and really bridging everything from clinical genetics and space medicine to computational biology and synthetic biology. I had the pleasure of serving as one of the members of the steering committee for Gene Lab way back when it was before OSDR became the Open Science Data Repository. So I've been working on some level with the working groups and OSDR since the early days of Gene Lab back in 2018, 2017, and been really excited to join some of the working groups like the multiomics working group that's led by Dr. Beheshti, microbial working group, some of the more even exploratory adventures in the plant working group. So it was a lot of great working groups that get students involved in data analysis and papers and new ideas.
Yeah, so I used to be at NASA Ames Research Center before I was at the University of Pittsburgh in September. And my first position there was to work on what used to be called Gene Lab, right? OSDR was Gene Lab. And that's basically, that was back in, was it 9 years ago now? 10 years ago? I can't even remember. Time flies. But yeah, that was around there. And that's when, yeah, obviously I was helping develop the platform and with Dr. Sylvan Koss, of course, leading the charge as the project manager then. And yeah, and then the multiomics AWGs were formed because Sylvan was the person who was initially came up with the idea, oh, we should have these kind of AWGs analysis working groups to have the scientific communities come together first as a way to assess standard pipelines for omics that the community agrees to. But of course, when scientists get together, we want to work on science, so that's where all the, you know, open science stuff came about then and evolved to what it is now.
We've had a number of really good collaborations with the working groups, and so this led to a lot of work that was published actually in summer of 24. This large package building on a lot of the Inspiration 4 data set, the first all civilian crew that was led by Jared Isaacman and the team there. So really, a really exploratory mission that was announced and planned and trained and all within about 4 or 5 months. So really exciting to get that together. And the working groups, we used a lot of that data across the multiomics working groups, some of the animal working groups, microbial working groups. So we were able to take a lot of the data, and once it was completed for the generation of the data, have that be a real nice, rich bed of raw data that we could then compress and analyze and compare to other data sets. And that was really what led to, I think, a lot of collaborations with those group, and eventually, of course, dozens of papers that came out of that large scale collaboration with OSDR and working group teams.
Early on, like some of the idea was how do we get the community to think how to use Gene Lab back then, now OSDR, to use it to come up with publications. So some of the first publications we came up with, what is it now, 10 years or 9 years ago, was with, some of the AWG members at the time was smaller. So then we said, how do we get papers together? So we worked together to get some papers out. And eventually we got the first paper package that Chris and I put out was a Cell Press. And that's where we realized as a community of the AWG, we could collaborate together and put out a high impact paper that ended up in Cell. That was the mitochondrial paper that came out of so the focus. So that was the key there, where the community started realizing, oh, if we work together as a large collaborative group, get rid of the boundaries, and then we could come up with neat papers. And then now it's evolved to what Chris said in the newer paper package that there was many papers that we worked together with collaborators from all around the world as part of our multi-omics group and other AWGs that worked on many nice papers from different variety of journals and subjects. So we covered whole lot of different subjects in space that wouldn't have been possible without the AWGs.
Being a part of the AWGs was a really great ecosystem of people with ideas or different algorithms or comparison data sets that they had. And so I think it opened the door to a lot of new ideas and collaborations. And also, what was nice that it had a good, most meetings have a monthly cadence. So you'd think about data presented, get feedback, and would be able to come back to a group of peers and colleagues that had also been looking at some of the same data for a month or so. So it was really great to have this continual iterative feedback about figures, analyses, questions, best practices, and data cleaning methods, really everything.
Yeah, I think that my contribution, I mean, granted, I was leading the multi-omics group, so that I was trying to guide projects forward. That was something since leading the multi-omics as part of my job to be like, okay, how do we get projects facilitated to push forward and people to work together? And then some of the more junior people who are coming up with the ideas, how to promote their ideas. So from graduate students to postdocs to junior faculty that had ideas. So I said, as Chris said, yeah, they would present monthly in our meetings the ideas, and then you would get the group of people who are interested to work on that project came together, and then they started working on that project together, and then I would help push that project along a lot of times.
The working groups are a really great kind of call to action, army of scientific concepts and papers and data. So it was great.
It's really great that the
Yeah,
whole
that's great.
community is contributing from it, all corners of the world. So not just the U.S. and Europe, but it's everywhere. So the Deep Space Countermeasure package was our next venture of onslaught of papers to uh, come out to the public. So Chris and I have approached Cell Press about a deep space countermeasure package, which involves anything related, as it sounds like, deep space and countermeasure. And the whole goal is to idea is, when these paper packages come out, you actually see the pieces of the puzzles pieced together. Like, it's harder to piece things together when you read two papers that are 3 months apart because you might have forgotten half the things that were in the other paper. But when the paper packages come out, you actually see everything together at once, and you can piece the puzzle together. Or our idea is to figure out with this large amount of papers coming out in this package is have a countermeasure atlas in a sense. So now you can have this countermeasure atlas there for the first time of all the countermeasures that are people working around worldwide in one place. So then you could actually see, oh, maybe let's say one paper's working on a mitochondrial supplement, which I'm working on and Chris is working on, but another paper's working on some other kind of supplement or drug. And these individual papers, you read them, you're like, oh, actually these two countermeasures might be synergistic together. So now you figured out a way to make a cocktail and then make the optimal countermeasure. So that magic pill that you see in sci-fi movies, the person pops in, oh, I'm cured from space. It's the goal for me. This package is that's the goal to really create that atlas. And then since we're going to deep space, there's a lot of Chris could talk about all the deep space stuff since he's really pushing that part of it.
Yeah, and absolutely. So this it's kind of like a catalog of places where places, tools, and methods to help us survive in deep space. And obviously, a lot of them are pharmacological, but there's other ways to help protect the body. Everything from looking at model organisms to tardigrade data to microbes that are really radio resistant. So there's a lot of great exemplars from nature and from chemistry that we can use to better understand the body's response to deep space and radiation. And some of the papers also look at, yeast that have been flown as far out as the moon and back. So there's a lot of interesting papers that start to actually get into deep space, which technically is supposed to be beyond the moon. But everything really between the Earth and the moon we know is a higher radiation risk. And so we really want to start to prepare for these missions that are already announced that will have a lot more traffic between Earth and the moon and Earth and Mars and maybe as soon as a few years. So it's not that far away. And we really don't have a good armamentarium of countermeasures that we can deploy, nor many ways to combine them that have yet been published or characterized. So, yeah, so this new package is to focus a lot on the deep space questions, deep space countermeasures, and just in general preparing us to go for this kind of next epoch of deeper space exploration. The a really great thing about OSDR is it houses so much of the data, almost anything that's been in space. If it has some omics data, is there. So there's, of course, there's rodent data, cell lines, plants, bacteria, yeah, and fruit flies. There's even some variations of derived cells and organoids. Almost everything that you want is up there, and also data processing tools and download tools. It's also actually from last year we managed to get the OSDR added as an official repository for Nature, so it's equivalent to NCBI or dbGaP or these places where you can store data and have a repository across all kingdoms of life, which is very unique. And it's great for code sharing, for quickly checking on a sample, linking to scripts, everything from thinking about an experiment to start to even process it and get some of the data.
Yeah, and I think part of it too is even the samples, the tissues, right? They're now on the bigger OSDR platform. So you can even deposit tissues. You can get tissues from people who don't have access to this stuff. So there's a lot of things that are brewing that I think are nice with OSDR that are going to help the community at large that want to get involved that don't have any funding to work in space or don't have any access to the tissues or samples. I mean, there's many surprises. I mean, the people are always fun to surprises because you never know when, I've been surprised by like even the junior folks, especially like from high school to undergrads who are actually participating in our AWGs and but the younger ones keep us energized. So then, that's the part where it's neat, whereas you see high school kids participate and then I encourage them to be part of papers and they've been part of some of my papers and they come up with some ideas. And even the undergrads and graduate students just lead some of the topics because I've been approached, for example, from undergrads and graduate students that say, "Oh, I have this idea. Is it doable? Does it make sense? Is it silly?" I was like, "No," and they come up with some really nice, novel questions, and that's a neat thing that the WGs can promote.
Yeah, and I agree. I think that, the people what's been really wonderful all over the world, people with different backgrounds, different ideas, different concepts of what they want to do for a research question. So it's been really, really wonderful seeing, just all these different kinds of people from around the world with different backgrounds, expertise, ideas, and yeah, many of them become collaborators for life.
The value of being part of the AWGs and the and OSDR community is that you get access to space data, obviously. Space tissue samples that most people don't have access to on this. This actually gives you access to free data, free samples that are out there that you could request from the OSDR site. Or with the larger community, for example, I have tissues that from my sent to space, in my freezers from previous grants and funds, and I share that with the community. If someone says, "Oh, I need a tissue of that," I'm like, "Oh, I have one." So and then I collaborate. You sign the MTAs and you send over the tissues, and then they can use it, and we could all benefit together. And plus, some people I know have approached me to join. It's like, well, what I do does that apply to space? They want to get involved, but they don't know. Let's say what they're working on applies to this, and we say, yeah, everything you do applies to space. So
Mhm.
that's where they learn everything applies to space, and then how do they do it? They work with people like Chris and other folks and myself and then say, okay, this is how our knowledge and techniques can apply to space research. So
Yeah, yeah, I mean, I think the best analogy would be so the biggest advantage of joining an AWG is really imagine if you go into a research laboratory, a big building, for example, and you might set up a bench and you could see the people next to you or people on the other side of the hallway. You'll see people nearby you, but imagine if you could suddenly walk into a large research building and have access and feel a camaraderie with everybody in the building all at once. It has that kind of sense of scale of connectivity that you get really quickly. Yeah, and people are very collaborative. There's also it's a culture that's I think been really well engineered by NASA to say this is a collaborative community. These are places where people can come up with ideas and work with each other. So it's a collaborative and instant scientific network. Last thoughts might be the AWs are very unique because it is a broad spectrum of very old to very young to mid-career scientists, engineers, computational biologists, people from all backgrounds really coming in. So it's really exciting to see and well, should be celebrated. It's a pretty unique place.
yeah, no, I agree. So I think that's definitely the benefit of it, where you have people from all around, and it's not just the career level, but all around the world too. So, for example, there's people from Australia, Japan, from in the U.S. People may not be able to interact with them because we're not at the same meeting. So, you get to, as Chris said, you kind of uh, walk into this basically large institution, you could call it, of many different disciplines and backgrounds. And if they say you need help from, oh, I need expertise in, looking at some protein or some molecule, you'll find it in the WG because it's such a large community and you just have to think outside the box.